Suchishmita. So let us start. It's already 11:31.
Yeah.
So with sir permission, we can start.
Yes.
Okay, sir.
Am I audible? I mean, can you hear me, ma'am?
Yes.
Okay, so I'll start. Thank you, sir. We'll start now.
Yes,
Very
yes.
good morning to all of you. Respected sir, our honorable guest today, respected principal madam, all dear participants, my seniors, my colleagues, and my dear students. I, Dr. Suchishmita Das from the Department of Microbiology of THK Jain College, welcomes you all to the webinar on COVID-19 invasion, a challenge to humanity. I hope you all are doing well in this current pandemic situation. We're still locked at home. We cannot go to our college, to university, cannot meet our colleagues, our friends. But let us all be very positive that this phase will be gone very soon and we can all get back to our normal life. So let's hope for the best, right? Today we feel very privileged that we have among us one of the eminent academicians of the country, Professor Dr. Dhrubajyoti Chattopadhyay, Honorable Vice Chancellor, Sister Nivedita University. For all his students, he is not only our favorite teacher, but he is like a father-like figure to us. Whenever we approach him, he is always there to give his valuable suggestions. Doesn't matter how busy he is, but he is always there. And with me, this has happened many, many times. Thank you, sir, for readily accepting our request. In spite of you having such a busy schedule, we are really honored. Thank you, sir. Today is also a very special day. Today is the 156th birth anniversary of Sir Ashutosh Mukherjee, and we all know his immense contribution for education, for society. So today we have to remember him. We'll be paying a small tribute to him. Thank you all dear participants for showing such huge response, obviously because DGC sir is there, is the speaker, our honorable guest today. And all the young students who are attending the webinar today and never got the chance to listen to sir's lecture earlier will have a wonderful experience. So all the very best to all of you. Now before we move on to our next session, few humble requests to all of you. I mean, I know you all know them, but it's my responsibility to keep repeating them again and again. So please keep your microphone and camera in the off mode, and please do not share or present any screen. So thank you, all of you. Now I'd like to request, we'll move on to the welcome address session. So with this, I'd like to request our respected principal madam, Dr. Moshmi Singh Sengupta, to give her welcome address and also introduce our respected speaker. Ma'am?
Thank you, Suchishmita, for your nice introduction. A very good morning to all of you. On behalf of the College Authority and Managing Committee of Taradevi Haratchand Kankaria Jain College, which is popularly known as THK Jain College, I offer my hearty welcome to all the participants of today's webinar on the topic COVID-19 Invasion, a challenge to humanity, which is organized by the Department of Microbiology. I accord my heartiest welcome to Professor Dhrubajyoti Chattopadhyay, Honorable Vice Chancellor of Sister Nivedita University. Many of us, especially those who are associated with the prestigious University of Calcutta in different capacities, know that 29 June is a very special day in the history of University of Calcutta, as it is a birthday of Sir Ashutosh Mukherjee, a man of academic skills, self-esteem and courage, who was the second Vice Chancellor of Indian Vice Chancellor of Calcutta University and held that position for 5 terms. This year, it is his 156th birth anniversary. I take this opportunity to express my sincere thanks and gratitude to Professor Dr. Dhrubajyoti Chattopadhyay for giving his kind consent to speak for all of us on this auspicious day. It is a privilege for me to introduce to our esteemed speaker, who is also my teacher, although he is someone who does not require any introduction. Professor Dr. Dhrubajyoti Chattopadhyay is a stalwart in the field of biochemistry, microbiology, and biotechnology. He is not only an illustrious scientist and administrator, but an institution in himself when it comes to biological sciences. He was the founder, Vice Chancellor of Amity University, Kolkata, During the tenure of his association with Calcutta University, he acted as Pro-Vice Chancellor for Academic Affairs, Dean of Science. He was the director at the Center of our Research in Nanoscience and Nanotechnology, University of Calcutta. Professor Chattopadhyay has enjoyed both national and international recognition for notable contributions in different areas of biological science. He was elected as Fellow of the National Academy of Sciences, Fellow of the Indian Academy of Sciences, and Fellow of West Bengal Academy of Science and Technology. Obtained their PhD under his direct supervision, and 10 more students worked under his joint supervision. He has around 130 research papers and 6 book chapters to his credit till date. I'm sure that many participants who are attending this seminar have been his students, and we know him as our revered DJC sir. He is a man of distinct vision, an idol of knowledge and experience. He is an inspiration to all of us. I'm very grateful to sir for taking out time from his busy schedule to enlighten the participants of today's webinar on a very relevant topic. I'm sure that today's session will be intellectually stimulating and enriching for all of us. Thank you, sir.
Thank you, madam. Yes, sir. Thank you so very much again for your consent.
Yes, I'm here.
Yeah, can you share the screen and start paying tribute to Sir Ashutosh Mukherjee? Thank you.
Is my screen visible?
It's coming. It's there. Yeah, it's coming. Yes, it's coming.
Thank you. Yeah, thank you so much. Good. Thank you so much.
Yeah, it's there. Yeah, it's coming. Yes, it's coming.
Thank you. Yeah, thank you so much.
Thank you.
Good morning to the respected resource persons, sir, principal ma'am, faculty members, and all other participants of this webinar. I am Professor Nivedita, faculty at the Department of English, P.H. Kejan College, Kolkata, and I will present the PowerPoint presentation to commemorate the birth anniversary of Sir Ashutosh Mukherjee. The 19th century in Bengal was deeply and thoroughly influenced by the social reform movement known as the Bengal Renaissance. And it was during this time this was born. Sir Chattopadhyay, as he was popularly called in Bengali circles, Bangla Bagh, or the Tiger of Bengal, a nickname that fell his book. Scholars, doctors, engineers, Sir Ashutosh grew up in an atmosphere of science and literature. Under the guidance of his father, he develops the value of discipline and a quest for knowledge. This glorious meaning, coupled with his own personality and a tenacious melody, captured in Gaelic goods. It was also during this time Dhrubajyoti was a pioneer of the banking system. We can call this a chance meeting between two illustrious Indian educationists of the 19th century who would become the greatest Indian educationists of the 20th century. This had immense impact on Sir Ashutosh. That early brilliance which Sir Ashutosh had in his academic pursuits continued throughout his of the first student to be awarded a dual degree, MA Mathematics and C in Physics from the University of Calcutta. Here, a glance of Sir Ashutosh's achievement in of mathematics. Past research papers, mathematics being published Library Messenger of Mathematics. He was examiner at the edition in mathematics just the year after he received his master's degree. Sir Ashutosh next turned to law and there his brilliance continued. Several law degrees, enrolled as a lawyer in the Calcutta High Court, a professor of law, and finally a judge of the High Court from 1904 to 1823. To some of his glorious achievements, served 5 terms as Vice Chancellor of the University of Calcutta. He was a capable administrator. We also look at his role in the Hazra Law College, the Calcutta Mathematical Society, Ashutosh in the Science Congress, and others. Ashutosh now using the immense administrative experience of the Vice Chancellor of the University of Calcutta and also Judge High Court for his educational ventures. His contributions become a testimony to his original role as an Indian educationist.
Good morning, everybody. It is really indeed a great pleasure for me to interact with a large number of my previous students, and I'm so happy to see all of them over here, and almost the microbiology department of all the different colleges are being represented, and thereby I really thank the extraordinary effort taken by the principal, Professor Moushumi Singh, as well as Sengupto, as well as Shushishmita. Both of you have done a real great job, and I feel so happy to see everybody over here. And I could see that most of the colleges where I actually being instrumental in starting the microbiology department, many of these colleges are being represented over here. So thank you all my different colleagues from different colleges, and thanks to all the faculty members and colleagues and staff members of THK Jain College. In fact, I feel very proud that when I started, actually, this particular department being started after I actually visited the college and given this recommendation of starting the Department of Microbiology. And that has been developed in such a big way. It is one of the very important things for me. Start because today is Sir Ashutosh Mukherjee's birthday. I just want to, one of the very close to my heart, one of the Ashit Barun Giri's tribute, I just want to say that, an ode on a titan. Sonnets are written anon for one. Songs are sung oftentimes for one. Floral tributes are indeed laid for one. Encomiums are crafted for one. Books reverently penned for one. Speeches are delivered for one. Retrospectives are religiously held for one who lived in deeds and not in years span. And that's why Sir Ashutosh Mukherjee is still a person who is alive in all our mind, in all our other very important Bengali person, Prashant Mohalanavish, and that is also important for us because both of them played very important role. Today, when Moushumi and Shuchishmita requested me to deliver the lecture on COVID to my students and others, uh, the COVID-19, though we looked at it, damaged quite a bit of things. I looked at it from a completely positive side. I looked at it from understand quite front of a test at home period of lockdown. So, the last couple of years, almost I'm not reading any books. I just looked at the introduction part or some parts, but the novels, almost I'm not reading anything. But during this particular period of time, I read all the virus, epidemics, and other things. So I just actually giving you some idea about the books I have gone through. All of these are available in the Amazon. And it's so interesting to find out this pandemic situation and looked up by scientists, writers, the academicians from so many different novelists, so many different angles. Here are the examples of a couple of books on virus, on the dreadful activities, on the pandemic, on this particular, you can see this Ebola, the vaccine race, which is going on right now, richest restaurants crisis red zone, the fatal fever, the influenza, the more deadly than, and the infected cell reads the RNA, and getting actually, you can get the making proteins that will keep the immune system at bay and help assemble the new copies of the virus. The infection process, new copies of viruses are there, and they are actually coming out of the cell. On the other hand, our own immune system, they releases antigen-presenting cells or APC. And these antigen-presenting cells, they engulf the virus and display the virus peptides, which allow other immune cells to recognize the virus too. And the viral peptide, the T helper cells latch onto these viral peptides that have been flagged by the APCs and help activate other immune cells like the B cells and the cytotoxic T cells. And the B cells, they produce antibodies that help prevent the virus from infecting cells, and they can be long-lived memory B cells and T cells that recognize the virus can patrol the body for months, providing immunity. On the other hand, cytotoxic T cells identify and destroy the infected cells. So that is how our body at the initial stage can fight out the virus. But if they cannot fight out the virus, then naturally you can see the different organs which have been affected. And here I have shown the different organs. Next slide. And there is a putative association between severe COVID-19 and the indicated pre-existing chronic diseases. You can see that pre-existing chronic diseases always be said that hypertension, the chronic kidney diseases, then the liver injury, then the diabetes, all these are actually the one which has got a correlation between the severe COVID-19 and the pre-existing chronic diseases. So you can see that the pre-existing chronic, that's why we said that these are the people whom we have to keep very, very isolated so that they cannot get infected. Next slide, please.
Sir, can I make a request? The slides are not visible in the YouTube live streaming. Somehow it is not there. Shunima, can you start screening again? It is here, but YouTube it.
The pre-existing diseases, I already gave you the correlation between that. If you have hypertension, if you have diabetes, if you have kidney function disorders, or if you have liver function disorders, in these particular cases, there are more fatal cases out there. And now also people are looking for these epigenetic factors, which also there are some important publications started to come up. Next. So the COVID-19 fatality risk, just I have mentioned to you over here, and there this is actually we could find it out the low risk patients and the high risk patients, where the cytokine storm is actually being responsible for these high risk patients, and thereby it is found that there are epigenetic dysfunctions are also there. The immune sequence, inflammation, biological age, and the environmental factors, comorbidities are very important. So epigenetic dysregulation of the immune system and the renin-angiotensin system may increase fatality risk. A variety of biological clocks have been shown to predict the health and the longevity more accurately than chronological age. And individually, the biological age greater than their chronological age is thought to be undergoing accelerated aging, which may increase the risk of COVID-19. And thereby what I can say that these are being contributed by cardiovascular diseases, diabetes, obesity, COPD, they are at greater risk. So conversely, individuals who live healthy lifestyles, consume geoprotectors, protectors such as metformin, the resveratrol, NAD boosters may have decreased risk of fatality. Next slide. And this is actually the epigenetic mechanism so far identified in case of the search susceptibility, and you can see that how actually the something I think it's a very important area to be researched. Next. Looking at this common thing, I am not going to this very in details. This is a comparative analysis of COVID-19 case fatality rates by country, sex and ages. And this clearly shows that what is actually the discrimination between this. Next slide. Over here, this is actually known the sex differences that may impact immune responses to SARS-CoV-2 and COVID-19 progression. And hereby, as I mentioned to you, there are certain roles of the sex hormones also. Next. Next. So, COVID-19 disproportionately affect the older people. And if you do look at this, the upper respiratory tract organization of the the aged people and the youth system, you can see the difference between the two. This has clearly been shown over here. And the aged system, the viral alert signals are initially slow, resulting in greater viral replication. Defective macrophages and T cells, the limited repertoire of receptors, are less effective. More cells are infected, inducing high levels of inflammatory cytokine signaling. And the endothelial cell lining of the capillary becomes inflamed. Fibroblasts are activated and SARS-CoV-2 viral components and cytokines enter the bloodstream. Fluid fills the alveoli, reducing the lung capacity, and the virus infects microvascular pericytes in other organs. And the cytokine storm infects microvascular clotting, causing severe hypoxia, coagulopathy, and organ failure. The CPGs in the ACE2 promoter decreases with age, and these CPGs are bordered by long-range promoter enhancer contacts that may change over time. Similarly, it was found that in the population studies that there is another enzyme called ACE2 which acts as a regulator for ACE2 expression, and the ACE1 actually represses the maximum expression of ACE2. ACE2 is also very important for our normal activities, and that actually being regulated by ACE1. Now, in the population where the ACE1 is not present or ACE1 expression is less, then the ACE2 get dysregulated and there will be a high production of ACE2. High production of ACE2 means there will be more infection, and in general the Caucasian population has got similar type of things. Next. So in general, the age-related changes that increase the COVID-19 susceptibility is shown over here, and I have just discussed with you, and you can see the different points, different parameters which have become very, very important. And I can tell you that there are areas of research going on which is giving us more and more information regarding the dysregulation of AC2 receptor expression, which actually helps in the production of more receptor. More receptor means more infection and fatality. Now, naturally, a comparison of this COVID-19 tests which are available right now, all of you know the whole market is filled up with. One is a rapid test, is a real-time RT-PCR test, and another is a rapid antibody test. But you can see that when I discuss about the life cycle, immune cells are only going to appear after some time, after this infection started to produce the proteins, new virus particles, the viral peptides are there, and then only it gets the immune system starts to function. So this IgG and the IgA and the IgG, they started to appear later in the infection. Sometimes it after 7 to 8 days and 10 to 12 days. So when these antibodies are going to be there, then only the rapid antibody test can be taken. So rapid antibody test can be taken that if the population has got a large number of population has got this antibody, that means the immunization has already been taken place naturally, and that's what we call as a herd immunity. But you can easily understand the herd immunity should not be an option for India with 1.34 billion people. And this particular number of people who are getting more affected by this is going to be impossible. So the antibody test is effective for whom, and the RT-PCR test, both of these are actually shown over here. Next. And so basically there are different labs which have came out with this, but the Delhi IIT one and the GCC Biotech, they are also producing now. The GCC Biotech already got some approval, and which is actually from one of the South Diamond, close to Diamond Harbour, company which is producing this, and the other one which is actually from Delhi, this one, which is right now is still not available in the market, but will be available in the market in a very short period of time. Next. So it all depends on challenges, it depends on the number of test kits available, quality of the test kits, because you know there are a number of test kits, and you can see in the newspaper already that they have been tested first negative, then found to be positive, or positive, then found to be negative. All the different types of differential test results are also sometimes a big challenge. The capacity of testing labs and the compliance with government guidelines, and you can see this capacity is increasing every day. Right now a large number of testing is going on in India, and the shortage of safety gear for health workers involved in testing is also very important. This is also being considered as a challenge. Next. And this is another one which is also not, I think within a week it will be used very much. This is also done by the IIT scientists in Delhi, and they called as the Cas9 based editor-linked uniform detection assay, or FELUDA, and that FELUDA detects whether you are gets the infection or not. It is a CRISPR-Cas based system, and hopefully this can give a very, very authentic result also. This was also done by Bengali scientists. Next. So in general, what we could see over here, that patients with this consistent clinical pictures like the fever, cough, and this particular difficulties in the respiratory system, and the positive CT scan, they are actually going to be considered as the one. and the exposure to the epidemic areas, contact with a known case are also going to be considered. And if you can see, it's yes or no, there are two different types of activities to be done. And this actually gives us an idea that symptomatic and asymptomatic patients, asymptomatic numbers are much higher, and asymptomatic numbers are actually the one which are dangerous because we don't know, because they don't have any symptoms, because they have the sufficient system to combat with this virus. So asymptomatic can be divided into three stages. One is they're not at all infected, another is mildly infected but controlled, another is fully infected. They are actually going to show the symptoms very soon. So this last stage is fine, but the first two cases are actually the one which is very difficult to differentiate. So there are a number of vaccine development proposals are already been there, and you can see that the different types of vaccines which initiate an immune response to detect and recognize a pathogen, and there are different types. One is called the virus vaccines, which are actually used for polio, salt and salmon vaccine, which either use the inactivated virus or the weakened virus. But you know, the inactivated weakened virus, these are already, there are common type, production capacities are largely available, and there is a collaboration going on between US-based Photogenics and the Serum Institute of India, and to get a weak coronavirus and use that for producing the vaccines. But you know that the risk is there. Amongst that, it can cause infection. The viral vector vaccines are of two types. One we call as a replicating vaccines, another is called the non-replicating vaccines. And these, say for example, the Ebola vaccine is a virus vector. So expertise required to produce this type of vaccines is available, and that can be used for COVID-19 vaccine development, and people are trying that. And any sort of existing immunity to the viral vector could make the replicating type less effective. The nucleic acid vaccines can also be a two-type DNA vaccine and the RNA vaccine. And right now we know that messenger RNA-based vaccine in the Oxford team they are doing, and there is another team, the Australian team, they are also doing this type of vaccines. And then we are expecting the results, and people are actually looking at what's going to happen. But the protein-based vaccines are becoming very, very more numbers. So there are 123 number of vaccine projects which are going on. Out of them, 10 are in very almost final stages. So there are protein subunit vaccines. There are 20 teams are working on this, and this focus on the virus's spike protein, or a key part of it, or the receptor binding domain. And virus-like particle vaccines, there are 5 teams which are working on that. These are proteins or protein shells that mimic the coronavirus outer coat are used. These antivirus shells are not infectious but can produce antigen. These vaccines are safer, easier to produce, and the SARS vaccine of this type protected the monkeys against infection, already demonstrated. And they also often require an adjuvant to boost the immune responses. And in certain cases we could find, next slide please, that antibody combinations are also there. But here I want to mention about one thing. Rabindranath Tagore long time back actually mentioned during this particular break of Spanish flu, and that is actually in his novel Chokhel Bali, where the Bihari, one of the character, is mentioning that it is better not to wait for the medicines to combat the viral disease. It is better to take precautions to have the viral disease. So the main thing what I can tell you, that this stay home, that prevention is much better than cure. So stay home as much as it can, keep a safe distance, wash hands often, cover your cough, and you know that you actually can do it and do it and do it, and make the people aware of this. There are a large number of people who are not aware, or thinks, oh, nothing will happen to me. Nothing may happen to you, but you can do something bad for the others. So we have to think that one also. Next slide. So this is actually the chronologically what the SARS-CoV infection is actually there, and you can see that these are actually how one after another is going on. I don't want to repeat this again. This is a little bit more detailed where interferons roles are also being mentioned. There are different types of interferons which are actually looked upon right now. The IL-6 and IL-10 are two candidates which are actually being looked upon. Next. In general, but it is possible to demonstrate that this is actually showing the chronological of events SARS-CoV-2 infection, and this is how we can look at the SARS-CoV-2 infection one after another and stage by stage. Looking at the people who are attending this, where there are a number of people who are not typically from the bioscience group, I just want to not to go through it more details, but just to let you know that these are the research areas which are going on, and in such a short time we got this research. So COVID-19 given us another very important lesson to do a fantastic research, to do a good research. Collaboration is very important within the country, outside the country. You know that many of these particular papers are coming out by collaboration between the different states of our country, different institutes of our country, different universities of our country. Many work is going on between the countries also. Large number of results are coming up jointly by different parts of the country. This is also another very important thing what COVID-19 actually tells us. So next slide please. So the potential therapeutic actually based on what I have said, that if I understand the life cycle better, it is possible to develop these particular therapeutic approaches against the SARS-CoV-2. You know, right now everybody is looking for the vaccine, but I'm not very sure that even if some vaccines comes early in end of September, October, whether that's going to be available for India or not. And also the efficacy of these vaccines are also sometimes you have to test this. Normally vaccine takes almost 1 to 2 years. These are because of this situation, these things are being done. But you know that sometimes, so I want that the first step that to avoid to have this infection to spread is much better. And then slowly, you know, the community starts actually understanding this much better and do things. The second, next slide. The second thing what I think this particular repurposing of the drugs are becoming very important. You know, the remdesivir is already being considered. This is a FDA approved drug for the Ebola virus infection, and this particular remdesivir was found to also decrease the rate actually of infection. So it may this particular you get a longer period of time before going to the stage 2, stage 3, so that more treatment could be done not to go to the that particular level. So the remdesivir specifically targets the key viral proteins normally, and involved in making the new copies of the virus present in them. And over here, you know, RNA is a genome, so it's a RNA-dependent RNA polymerase. So inhibitors of RNA-dependent RNA polymerase are becoming very important. Next slide. Similarly, it was found that this particular lopaniruvir, ritonavir, and bounds are also coming. The favipiravir, already I have mentioned about why the avian influenza virus antidote is effective in our SARS-CoV-2. I explained to that, and it was found that this particular molecule is going to be very important. And these particular molecules are already been produced by India, approved by FDA, and the prices are also less, so is affordable also. Next. So the chloroquine, you know that at the very early stages, there are some reports coming up. And normally what happens, you know, that this chloroquine, if you remember, after this infection, it goes into the endosome, where the pH goes up and the pH goes down, the acidification opens up this RNA to come out of the virus particle and gets into the cytoplasm. But if that endosome's pH is increased by hydroxychloroquine or chloroquine treatment, then what will happen? The virus cannot actually get this RNA out, and in that way it can. But now it's a very optimal. The amount of chloroquine, hydroxychloroquine, it was found is very much susceptible to that. And now, really in different parts of the globe, this use of hydroxychloroquine has been banned. Next. And there are potential antiviral drugs, as I mentioned already. Next. Recently, it was found that a non-competitive pair of human neutralizing antibodies, you remember that the last stage where the B cells and the T cells started to produce the antibodies, they block COVID-19 virus binding to its receptor ACE2. So, and there is another paper in Nature, development of an inactivated vaccine candidate for SARS-CoV-2. Now, the inactivated vaccine candidates are always been looked upon in little bit pinch of salt, and that's why these human neutralizing bodies, which are actually done by the Israeli scientists, are actually going to be very important also. Next. The treatment of these critically ill patients with COVID-19, the convalescent plasma is also another. But you know that convalescent plasma having the antibodies to the level, and this is not going to have cross reaction. There are a lot of things which has to be looked upon, but good results already started to come up. One of the central minister receiving cured, and in different places already it's being started, but it is not going to be a very common practice. So absolutely terminal patients, we have to do this. Next. Now this is actually how I mentioned to you at the last stage, the B cells produce antibodies, and this is actually not the innate immunity, which actually first defective immunity, which is actually the one which develops in the system. Right now, it is found these antibodies' life is also not very long. And within 3 to 4 months, these particular antibodies are actually going out of the system. So that is also a difficult thing. And that may be one of the reasons all of us are actually looking for a resurgence of this virus. Because you may have the infection cured off, you have the antibody, but that will be there for 3 to 4 months and again getting infected after 4 months because you don't have the antibody. So this is another very difficult situation. Next. So this is actually the one which shows the host response to the SARS-CoV-2. And in comparison to other respiratory viruses, SARS-CoV-2 infection drives a lower antiviral transcriptional response that is marked by lower interferon 1 and interferon 3 levels and elevated chemokine expression, which could explain the pro-inflammatory disease state associated with COVID-19. This pro-inflammatory state is then converted to the hyperinflammatory in the stage 3. Next. And these are also another very important point to look after this COVID-2 infection. People already started that how it changes the translation. I have told you about the epigenetic changes. I have told you about this particular other changes, immune changes, but the translation machinery of the host is also being changed by this particular SARS-CoV-2. And you can see there are a large number of things which are happening, and large number of hostile translation are being changed upon SARS-CoV-2 infection. And this potential outcomes of this antibody response to coronavirus is a very, very important thing which I already mentioned to you. Next slide. So this, as I mentioned to you, the pro-response leading to ultimately the hyperinflammatory response to cytokine storm is the complete story of this. I shall not go through in details, but this is one that you know that it has got pro-inflammatory followed by hyperinflammatory stages. Next. So these are the immune suppressants. So at that, if somebody goes to that stage, you need the immune suppressants to reduce that particular cytokine storm. Need immune suppression to treat the patients. Next. Next. Next slide. Next slide, please. So this is actually the what I can tell you, that this immune-based theory under evaluation of this treatment of COVID-19 been shown in this particular slide. You can see that using the interleukin 6 inhibitors, you need using the JAK genus kinase inhibitors, people are actually looking at, and in certain cases recommending that. These are the different types of things. In summary, I can give it to you. Next slide, please. And when we talk about this contribution of this hyperactivated monocytes, this contribution of this hyperactivated monocytes to coagulate in COVID-19 is again another type of response. And you have seen that a couple of weeks back, people are talking about that aspirin or something like that may be useful for this, though it has been negated right now. But they said because of this coagulation, it could be avoided and you can go ahead with it. Next. And this is actually the antithrombic therapy in patients with COVID-19. And I mentioned you that why this antithrombic therapeutics are also under consideration. And the way we are doing this is actually to give you all the microbiologists an impression that there are different angles which have been used to attack this particular infection and to get a cure out of it. Next. So almost I am at the end of this. Whatever we have discussed so far, because still we know very little about this infection, we don't have our weapon to fight against this war. What we have to do, we have to take more precautions. And that precautions, we have to think about. We should not have any racial stigma. Blaming a particular community for the current situation is not to be done. Violence against health workers, which is very common at the very first time. We have seen number of nurses, health workers, and medical persons, they are not being treated properly when they go back home. That is also have to be fought. And lastly, the affected individuals are facing a lot of social stigma. Oh, if that family has got one COVID patients, almost we are making them ocho. This particular social stigma is also dangerous. We have to understand that being kind to the affected individuals. So you have to take precautions, but you do not show any social stigma against a family, against a person. Because it has got so much of stress developed in that particular family, you can easily understand. It depends on facts and not rumors. There are huge amount of rumors going on. We have to understand the facts, spread the awareness regarding the do's and don'ts as endorsed by the health workers, the governments and others, and providing the mental health care. This is very, very important. The mental health care for all of us is very important, because these are completely a new situation for all of us. And in that way, we have to look at it from thing where we have to fight against our own and also fight against the external things. So these two fighting will definitely help us to fight out the corona. And I just want to say that I must give the huge thanks to the healthcare workers on the front line of the coronavirus pandemic. Salute to them, and we have to understand that this is nothing but stay safe, stay home, stop coronavirus, and lastly, stay positive. This positive attitude is also very important for us. Thank you very much.
Sir, for such an elaborative and informative presentation. We all got enlightened. Actually, I went back to my MSc Density Biology class, sir. Sir, we'll now move on to yeah, we'll move on to our interactive session. There are a lot of
Yes.
questions. Just a minute, sir. This session will be taken charge by Dr. Shonima Talapatra Ghosh and Professor Sukhanti Bhattacharya. They will read out the questions. So, if you can, Shonima.
Sir, I'm
Yes.
there. Thank you, sir. So I'll just read out the questions for you. There are many
Yeah.
questions. First question, it is from Dr. Moitri Mondal. She has asked, sir, I have gone through a paper for few days back. They found T cells has been reduced in some COVID survivals. How it is happening?
Can you repeat the question?
Surely,
COVID?
sir.
Yeah.
She has gone through a paper a few days back, and it mentioned that T cells has been reduced in some of the COVID survivals.
Yeah.
Now, how
Yeah.
it is happening?
So if you can see that if I told you, I mentioned to you that when a person goes into the stage 3, then what happens? The hyperinflammatory responses are there, and thereby there are large number of T cells being affected. And if that case happens, then what will happen? It actually makes the system, the T cell reduction system in the system, isn't it? Destruction of the T cells.
Okay. The second question from Dr. Gargi Shahakesh. She asked, is it possible that a person without any symptoms can suffer from severe headache and seizure all of a sudden and die within a few hours in corona disease?
Corona disease is not, this is a typical symptoms of corona disease. So a high fever followed by seizure, this must be something which is not related with corona.
Okay, thank you, sir.
It takes a time. A person gets infected, the virus replications and other things, and slowly how the host system is actually being captured. And you can see the symptoms. Those are important for this coronavirus disease.
Thank you, sir. There is another question from Aparajita Mukherjee. She is asking that many people are saying that the virus sits in throat for 4 days. Is guarding really helpful? Can patients with comorbidity be saved with nourishing food?
Yeah. So definitely the first question is actually, that as I mentioned to you, that it goes to the nose and the eyes and the mouth. So it passes to the throats, and you can have the cells adhered, or these particular places adhered with this. Now, what is actually what we are doing with the gargle and other things is actually washing the place. So it is actually the washing this particular thing is going to, because it actually helps the people to actually wash your system, and that definitely going to help. But this is not a concept is there that this particular temperature is going to kill the virus and others. So directly, if we look at the virus from outside, it can stay at 54 to 56 degrees centigrade for at least a certain period of time. And during gargle, we cannot keep that particular temperature in our body. So this is actually to wash out the thing. And the second is in comorbidity patients, the nutrient is very important, because nutrients is always helps in actually the supporting, making the system, immune system strengthened.
She has asked another question. Many people are not even using masks. How can we be saved with this increasing number of patients?
Yeah, that is what I said, that it is important for us to make the people in our surroundings aware of the fact that mask is must. Unless you need, you don't go out, but if you go out, you must have mask, and this mask is very, very important, because this particular person may be an asymptomatic person, this person may be in any one of the stages of asymptomatic. And then what will happen? Normally, by the max, what we generally do, that we shed when we sneeze, almost 40,000 virus comes out, 40,000 droplets comes out. When we cough, it is around 20,000. And when we just actually get the sputum out, that each droplets which are coming out of our system, if I'm asymptomatic, that may also contain the virus, and they are going to be dangerous. So max is actually going to be protect the others. Similarly, it can protect me also from the others. Actually convince them that everyone should have mask.
The next question is from Dr. Madhumita Moitra. She is asking, the cytokine storm is not experienced by many asymptomatic persons. Why? Does that mean?
Yeah, because asymptomatic person never goes to the stage 3. Once it goes to the stage 3, unless that you cannot hide this. So stage 1 is a virus multiplication, going on to the stage 2 when the pro-inflammation is converting into hyperinflammation, and this hyperinflammation actually results in the cytokine storm where all the chemokines becomes active. So unless you go to the stage 3, you cannot have that. Patients never goes to the stage 3. They combat it within stage 1.
Next part of our question is, does that mean that they will not have a cytokine storm in other respiratory viral infections?
No, it's a question of these particular cytokine storms depends on that it is a host response. So the host at that particular stages of infection, how it is actually reacting to this particular infection stage is very important. But in many of the cases, not only the viral disorders, in normally many other stages, the pro-inflammation changes to the hyperinflammation, it takes place. So cytokine storm is very prominent in the stage 3 of this because of the typical nature of its response to the interleukins.
So next
I tried to give you the interleukins which are involved and how it actually helped in.
Okay, thank you, sir. Next question is from Nirmalya Chakraborty. He is asking, can bacterial cell surface components play any role as immune modulators to prevent COVID-19? Since we know may bacillus species can produce antibiotic type of lipoproteins.
See, the immune modulators, role of immunomodulators are always important. But in this particular infection, it is actually our inherent immune system, how it is actually doing these things is very important. Now, once you have an infection, say for example, parallelly you have another immune modulator in the system. These two cannot act properly in conjunction with each other to actually react. But in certain cases, there are 1 or 2 papers I have seen where they have actually said about this thing, that immune modulators can actually help in this particular system.
Okay, thank you, sir. There is a question from YouTube link that has been asked by Orpita Moitro. If immunosuppressant is applied to inhibit the cytokine storm, how could it be useful?
It will be very useful, and in many cases it has been found that I mentioned in one of the slides which completely tells about the immune suppressions, and the immune suppressants then actually help in this at the last stage of infection.
Next question is from Shubham Bari. He has asked, how Coronil gonna help?
How?
Coronil gonna help?
That's not relevant question. Coronary is
Okay, next question is from Orpita Moitra again. How the loss of taste and smell is accompanied with the onset of infection?
Yeah. So, you know that I don't know why the government started to say that these are the new, but at the very onset of this infection, the symptoms of corona is always giving. See, I have shown you the list of the organs which are being affected by this coronavirus infection: the lungs, the liver, the brain, the neurological system. Now, if the neurological system affected, then what will happen? That will, at the very initial stage, that will actually our this particular taste buds and the other things are going to be affected. So that will be an effect on this infection.
Sir, another question from Jayashree Bhattacharya. She is asking, is it true that asymptomatic patients have less T cells?
Less?
T cells.
No.
T cells.
No. Asymptomatic patients, no, it's not less T cells. Asymptomatic patients actually have to restrict the virus at the stage 1. So it will not actually, so whereas the viral peptides are looked upon by the B cells and cytotoxic cells, and they are actually helping to produce the antibodies as well as destroying the infected cells.
So, shall I continue, sir, with the questions?
Yes.
Okay. So, another question from Anirban Shen. He is asking, does the remdesivir have any known side effects?
See, all these drugs has got side effects, but it is much less because remdesivir is being used for Ebola infection. It is FDA approved, and lot of actually research have been gone into this, and thereby the particular amount which has been prescribed actually has got very little side effect.
Okay, thank you, sir. Another question from one of our students, Abhishek Shaw. He is asking that whether plasma therapy or drugs like hydroxychloroquine or dexamethasone should be suggested. Which one is more effective?
Yeah. So you know that plasma therapy, as I mentioned to you, itself is number of tests and a lot of different things are going to be done for that. And it is not going to be available to everybody. It will be only for the terminal patients. There is no scope, and then we use it. But if you look at chloroquine, right now we banned. It is not really an efficient drug. Has been already been reported. Except in our country, all over the world, they are not using this. And over here also, the use is getting lesser. But dexamethasone is coming at a very, very potential drug, and dexamethasone price is less, and you can really treat with it. And in general, dexamethasone is actually being used for many people who are actually suffering from respiratory disorder, asthma, and others. So dexamethasone is a drug of choice.
Next question is from Saiva. The question is, what is the fate of the affected T cells?
So the fate of the affected T cells, as I mentioned to you, that these affected T cells actually going to, that's why what we call it's into the, it will try to produce viral peptides and antigen presenting cells, and these particular viral peptides are actually the T helper cells. They latch onto the viral peptides and activate the other immune cells like the B cells and cytotoxic T cells. And cytotoxic T cells, they identify and destroy the infected cells.
Next question is from Orpita Moitro. She is asking, if somehow a drug can be designed that can bind the ACE2 receptors, would it prevent the endocytosis of the virus? Will it reduce the infectivity?
Yeah. Before endocytosis, it will be effective itself, because ACE2 interaction with the spike protein, if that is prevented, then at the first stage, before fusion, you can actually prevent it. So people are trying to develop the drugs and design the drugs, and there are number of biocomputational efforts are there, and at least there are almost 150 potential molecules being identified, which are right now people are looking at, and to find out the efficacy as well as the affinity of these molecules towards this as a drug. So antiviral drugs is a potential area of research, and people are trying to do that. And that's why I appreciated that this is a first virus which right after identification within a month, its crystal structure of interaction with the host, the receptor as well as the spike protein interactive surface, and how this surface is interacting with each other. What sort of mutation will help less infection, which I have not mentioned. There are a number of different types of COVID right now could be identified, but there is one what we call as A2A, which is actually prevailed over in our place, is very infective one. And this particular, all these things are actually people are right now trying to get computer designed drugs to be developed.
Next question is from Dr. Priyanka De. She is asking, she has worked on cardiac hypertrophy model for DEX-induced heart, and dexamethasone has many side effects, especially on
Yes.
heart. So can
Yes.
that be suggested to babies?
Yeah. So, the doctor who is going to prescribe dexamethasone, they must have taken the patient's history. And dexamethasone is a much safer drug than anything else. So, the side effects where it can affect this particular babies or heart patients or others, this is not going to be done. But where it is possible to actually give the patient dexamethasone, it is right now the drug choice. His newspaper also has got a huge amount of information regarding the dexamethasone treatment.
Sharthak Mondal is asking, how does virus neutralization test works actually?
That it works in this way that if you have the antibodies circulating in your body, and when you are taking the blood out and try to find out that whether you have that antibody in the system, then you are going to do this particular test, and if you can identify the antibody which is circulating inside the system. See, normally what we could find it out that antigen circulation inside the system is very difficult in this particular case, but antibody detection by doing the reverse experimentation testing, it will be easier.
Thank you, sir. Shall I continue?
I don't know, it's already one.
Yes, sir. So, because there are many questions in the chat box.
Yeah, but I am sure that if the questions you can combine together and send it to me, I shall send the answer through email.
Okay. Thank you so much, sir, for your invaluable time. Now, over to Shuchishmita Di. Hello, Shuchishmita Di.
Chishmita, are you there? Thank you, sir. Sir, you have given us enough time for teaching again the virology, and really I can remember your classes, sir. Sir, you have explained it
That's my
so
love.
nice, so it's everything.
Yes, thank you very much.
So
Thank you very much.
now it's time to deliver the vote of thanks. Shuchishmita, are you there? Or I ask
Madam
Anamika,
Shuchishmita Di has left.
probably she has
Shuchishmita Di
left. Okay, I would like to request Dr. Anamika Ghotok of Department of Microbiology to deliver her vote of thanks. Anamika, are you there? Anamika? Okay.
Yes, madam. Am I audible?
Okay. Can you please raise your Can you please raise your voice? I think you check your microphone connection.
Am I audible, madam?
Yes, to some extent. Your voice is not very clear.
Okay.
Hello, Anamika.
Yes, madam. Yes. Am I
Yes.
audible?
Okay. You are audible.
Okay. Thank you, madam. Thank you, respected sir. A very good noon to all of you. At the end of this program, I deem it as a privilege to propose the vote of thanks for this webinar, COVID-19 Invasion, a challenge to humanity. First and foremost, I, on behalf of my institution, THK Joint College, would like to express our sincere gratitude to our eminent academician, Professor Dhrubajyoti Chattopadhyay, Honorable Vice Chancellor, Sister Nivedita University, Kolkata. Sir, despite his busy schedule, has found time to grace this event. He enlightened us with his very informative, explicit, and thought-provoking deliberations. Thank you, sir. My heartfelt thanks to Honorable Sri Sri S.S. Jain Sabha and governing body members of our college, Sri Satarma Kankaryaji, President, Sri Sohanraj Singh Ji, President of the governing body, Sri Lalit Kankaryaji, Secretary of the governing body, and other respected members for providing continuous support and encouragement to organize this webinar. I owe special gratitude to our respected Principal Madam, Dr. Moushmi Singh Sengupta, for her vision, leadership, and commitment. I would like to thank my fellow members of the organizing committee, Dr. Suchishmita Das, HOD of Microbiology Department, Dr. Shalima Dolabhajosh, Srimati Shemati Bhattacharya, for their endeavor throughout this event. We are grateful to all the teaching staffs of technical support team for their magnificent hard work to make this event technically safe and sound. Departments, Professor Undila Sengupta and Professor Dipayan Dor made a systematic presentation to pay tribute to Sir Ashutosh Mukhopadhyay for making this webinar meaningful. I am also very happy to convey our thankfulness to staff for their constant help. Last but not the least, we thank all the delivering faculty members, research scholars, students, and other participants present here accepting our invitation. Finally, we would like to give all the glory to the Almighty for making this event a resounding success. Once again, thank you all for being with us. Thank you.
Thank you very much. Thank you to everybody. Thank you, the principal, the governing body members, the faculty members, and everybody over here, the participants particularly, for their very interesting questions. And I really enjoyed the session. Namaskar.
Thank you, sir.
Thank you, sir. Sir, actually, I left the meeting because of the connectivity problem. I'm very sorry, sir. Thank you so very much, sir, for all your answers and explanations. We had a lovely time. And thank you. We'll again meet again. Thank you all participants. Thank you, Principal Madam. So the feedback link is already there in your email. So we'll be making it active very soon, just after the webinar ends. So kindly give your feedback and you'll get the certificates in few days. Thank you, sir. Thank you so very much for your time.
Thank you, sir.
Thank you. Thank you. Thank you. Thank you. Thank you. Thank you, everybody. Yeah.